This lecture is about the nervous system as a whole. It's made of the nerve cells that we went over in Lecture 1. And together, these will cooperate to carry out pretty much everything that matters in life. I think there's a classic philosophical question of if a tree falls in the forest and there isn't a nervous system around to perceive it, does it make a sound? We're not going to talk philosophy today, but we are going to talk divisions of the nervous system. So the entire nervous system is going to be made really of two parts. If you think of the nervous system as a whole, the first division would be central versus peripheral. The central nervous system is made of the brain and the spinal cord. So this is around the center of the body, hence the name central nervous system. And this is where we kind of process information, where we store information. The peripheral nervous system radiates off of the brain and spinal cord and interfaces with the body. And again, there are two divisions here. One is under our conscious awareness, that's the somatic nervous system, and the other one runs in the background. It's called the autonomic nervous system. It sounds an awful lot like automatic, and its function is an awful lot like automatic function. The autonomic nervous system is going to help maintain homeostatic functions. This is going to carry out fight or flight responses for the sympathetic nervous system, or rest and digest for the parasympathetic nervous system. The somatic nervous system is going to have both sensory and motor function, and this is what we're consciously aware of. The movements that we carry out, the feelings that we perceive, the sounds that you're hearing right now, these are conveyed via the somatic nervous system. The peripheral nervous system works together with the central nervous system to help provide sensory input and relay motor output. So these are all going to work together. The brain, for example, is going to come up with a plan and project that down via the corticospinal tract to the spinal cord. And then through the somatic nervous system, we then move our muscles. So I suppose we could draw another division here where we have our senses and where we have our motor function. And of course, all these arrows essentially go both ways. When we sense something, our somatic nervous system relays that to the spinal cord, which relays that to the brain. That sensory input is going to influence motor output as a result. So I like to think of the nervous system as a collection of pairs that all work together to really accomplish everything that matters in life. There's nothing that happens outside of our nervous system that matters to us. The only reason that we care about anything or have an emotional response to something is because of activity in our nervous system. So we'll start by going over the peripheral divisions, and then we'll go through the divisions of the central nervous system, mainly divisions in the cerebral cortex, since that's where the magic happens. So think of the central nervous system as the computer, the processor, the site of storage, and think of the peripheral nervous system as essentially a series of cables that are going to innervate the rest of the body. So right along the central axis of the body, we have the brain, the spinal cord, there's our central nervous system, and then we have these nerves radiating out. Spinal nerves are going to be a mixture of sensory and motor fibers, and as well as somatic and autonomic fibers as well. Also scattered throughout the body, we have little balls of cells called ganglia. So these would be collections of nerves that are going to play either sensory or motor functions depending on what they are. For example, the dorsal root ganglia, which are very close to the spinal cord, house our sensory neurons, somatosensory neurons. and the postganglionic neurons of the autonomic nervous system are also going to be found in the peripheral nervous system. In the central nervous system, we see divisions of gray matter and white matter. The gray matter is just cell bodies, dendrites, unmyelinated axons, synapses, and the white matter is myelinated axons, because myelin is made of fat. And so if you think about if you go to the store and buy a nice hunk of meat, there should be usually some marbling scattered throughout. So think of a nice steak. It might have a fat cap on it, but if you find a nice piece of meat, there's going to be speckles of fat throughout. That fat is white. The fat in our brain is white. It's no different. So the myelin found along those axons gives white matter its characteristic white coloration. And what you'll notice here is that in the brain we have gray matter on the outside, because the gray matter houses really the neurons and their synapses. It's where computations are carried out. So that's all on the outside. But the axons, which are connecting to other parts, the white matter, that's on the inside. As you move down into the brain stem and then eventually the spinal cord, this arrangement flips so that gray matter and white matter, gray matter being on the outside in the brain, white matter on the inside, it flips because the purpose of the spinal cord is to act more as a relay. So all of the descending and ascending tracks surround the neurons within the center. They carry out some fairly straightforward computations. So with the spinal cord, we put the white matter on the outside because the spinal cord is more of a relay. We put the gray matter on the outside in the brain because this is more of a site of storage and processing. Also scattered throughout our nervous system, we're going to have space that's filled up with cerebrospinal fluid. It's the fluid that we find in our cerebrum and spinal cord, hence the name. So this is just essentially a sugary saline solution that allows neurons to function properly. The larger spaces we'll call ventricles, and we'll go through those later on. The nerves that radiate off of our brainstem, for the most part, we call cranial nerves. There are two other cranial nerves that don't really hit the brainstem, but the other 10 do. The nerves that come off of the spinal cord are called spinal nerves. So these are going to innervate with the face and viscera for cranial nerves, and the spinal nerves are going to hit everything else. So body, also the viscera as well. We'll go through our cranial nerves in another date, but you should already be aware of the orderly arrangement of cranial nerves, where the first cranial nerve hits the olfactory bulbs. The second cranial nerve really just goes directly to the thalamus. But cranial nerves 3 through 12 arise from the midbrain down to the medulla in basically an orderly fashion. The spinal nerves also emerge in an orderly fashion as well. These are going to poke out in those intervertebral foramen, so the spaces between the vertebra. Here's the spinal cord. Notice the white matter on the outside. There are the dorsal roots and ventral roots. The ventral roots have motor function, dorsal sensory function. Coming out of these spaces between the vertebrae, we have our spinal nerves, and those that arise from more superior regions are going to innervate more superior regions of the body, and as we move down the spinal cord into the more lumbar and sacral regions, we're going to hit lower regions of the body. So it creates this orderly arrangement here. These spinal nerves are going to have a mix of somatic and autonomic fibers depending on where they arise from. The autonomic fibers will only arise from the thoracic, upper lumbar, and then sacral 2 to 4. So not every spinal nerve is going to have autonomic fibers in it, just most. That orderly arrangement and orderly innervation creates the dermatomes and myotomes that I'm sure you've heard of before. Here's an example of dermatomes. So the spinal nerves that arise from cervical regions are going to hit higher regions of the body. Thoracic is going to be down more near the trunk, a little bit of arm. The lumbar and sacral is going to handle the lower limbs. The gray matter of the spinal cord has some divisions in it. So the dorsal regions of the gray matter, this is going to be more sensory relays. The ventral portion is going to have motor functions. So somatic motor nuclei are going to be in the ventral horns. And then there are these lateral horns that have the preganglionic neurons for the autonomic nervous system. So here's another pair. Preganglionic live in the central nervous system, and the postganglionic neurons are out there in the periphery. So they create those ganglia that are scattered throughout. The preganglionic neurons are found in the lateral horns. Thoracic 1 to lumbar 3 for the sympathetic nervous system, and then sacral 2 to 4 for parasympathetic. Parasympathetic also arises from the brainstem as well. So it's sort of a parasympathetic a sympathetic sandwich with the parasympathetic bread. And we can see that sandwich here. So our autonomic nervous system, again, works in pairs. You can see over here, sympathetic, parasympathetic. So in the brainstem and sacral spinal cord, we have our parasympathetic fibers arising, and the sympathetic arise from thoracic down to lumbar. For sympathetic, think fight or flight. So this is priming the body for energy expenditure. Parasympathetic, rest and digest. This is priming us to store energy. And both branches are going to hit the same target organs, but they're going to have opposite effects. For example, at the bladder, the sympathetic nervous system is going to relax the bladder, so we're not excreting urine, whereas the parasympathetic nervous system is going to cause constriction of the bladder. We have opposite effects in the heart. The sympathetic nervous system is going to excite the heart, increase the heart rate, increase the force of contraction, whereas the parasympathetic nervous system is going to slow the heart rate. So at each target organ, one will act as the gas, one will act as the brake. The reason for that has to do with differences in the neurotransmitters that they release, which we'll get to in another lecture. Let's hit the central nervous system and look at some divisions and the basic tasks that are carried out. The logic here is that in the lower regions of the central nervous system, let's start us a new picture here. So here's our brain, got a little cerebellum. It's a bit ugly, but you get the point. Massive spinal cord down there. As you move from top to bottom in the nervous system, you go from higher order activities, like creating consciousness, for example, or creating some perception of color, and you move down to lower order functions, like simply contracting a muscle, or in the medulla, for example, breathing, swallowing. So as you move up, you increase the complexity of the task because you're essentially devoting additional neurons to it. It's all about location when we're talking damage of the nervous system. Where we damage it determines what happens. So here are the major divisions that we're going to go through. I'd say the more complex portions are going to be on the top and maybe a little simpler on the bottom. So the frontal lobes is where we tend to think of our consciousness being created. The frontal lobes are widely connected with the rest of our cortex, so they can take into account somatosensory, auditory, visual input, motor plans, emotions, and create that unified sense of now. Come up with a plan and then relay it to other parts of the cortex and, of course, down to the spinal cord. The parietal lobes, shown here in red, this is going to deal a lot more with somatosensation and visual spatial relationships. There can also be some mathematical computations that occur. It's a mixed bag. But when you're thinking parietal lobe, think somatosensation and visual spatial. For frontal lobe, think motor function, planning. In the temporal lobes, shown in green here, you should be thinking auditory function and, very importantly, memory and emotion. In the back, we have our occipital lobes. For that, just simply think vision. This is a very straightforward area of the brain. Visual input arrives here first after the thalamus. We make sense of what we're seeing at a very basic level and then pass that into different parts of the brain to figure out where is it. So we'll go to the parietal lobes for that. And what is it? We'll go to the temporal lobe. Because remember, this is an important site of memory storage. Covered up here, we have our brainstem with the nice thalamus on top. So here's our cerebrum, cerebral cortex that we were looking at before. Now if we peel that off, we can see the thalamus here. This is Grand Central Station. All roads lead to the thalamus, and the thalamus is widely connected with the cortex. There's one exception, and that's olfaction. What we smell actually goes to the cortex before it goes to the thalamus. Every other sense first goes to the thalamus, then the cortex. So the cortex gets very little primary information. For the most part, everything it knows comes from the thalamus. Below the thalamus, we have the midbrain. The midbrain sticks out because of this quadrigeminal plate here in the back, so your superior and inferior colliculi, and then of course the third and fourth cranial nerves coming off. So here's our optic nerve here. We got our oculomotor nerve and our trochlear nerve here. So 3 and 4 come off the midbrain. Second cranial nerve goes directly into the thalamus. So right below there we have the midbrain. Below the midbrain we have the pons. For the midbrain, you can get paralysis. You can have a loss of sensation. For the midbrain, we should really be thinking of this as a more basic relay. The thalamus can actually perform some computations, but the midbrain just contains a bunch of really important tracts. One of them would be our ascending arousal system. So this keeps our cortex stimulated enough that we remain awake. When this is damaged, we enter some sort of coma or a minimally conscious state. The midbrain also contains our corticospinal tract, so we can create paralysis, of course, by damaging that. And it also has a few special sets of neurons. Those in cranial nerves 3 and 4, so moving our eyes, but also dopamine neurons that are going to be very important when we talk about Parkinson's disease. Below the midbrain, we have the pons. This is another very important relay because it communicates directly with the cerebellum, which coordinates our movements. So damage in the pons can affect our ability to move. There's also a few pattern generators there that help coordinate activities like swallowing, so that we don't try to swallow and breathe at the same time. The cerebellum, which is just the little brain, as the name means, actually contains more neurons than the cerebrum because it's involved with a very important task, and that is coordinating our movement. So motor learning occurs in the cerebellum. At the bottom of our brain, we have the medulla. This area carries out the most vital functions: breathing, cardiovascular function, regulating our blood pressure, also swallowing as well. So vital functions are carried out in the medulla. Damage in the brain stem that affects medulla function runs the risk of perturbing respiration, which of course never ends well. So vital functions are carried out down here at the bottom of the brainstem. Higher order functions are going to be carried out up in the cortex. Now, in the cortex, we have many different functions carried out, and we can broadly create another pair here called primary and secondary cortices, or primary and association cortices. Same thing. The primary cortices, for example, this primary visual cortex, this gets direct input from the thalamus, so this is as close to primary input as we'll expect to get in the cortex. This deals only with visual information and with very granular bits of visual information. What is the angle of the line? Do I have any green light at all, any blue light, any red light? We're not figuring out hues in the primary visual cortex. That's going to be later on. So in association cortices, that's where we start to put together multiple bits of information. So we put together multiple lines, for example, to make shapes. And we can put together shapes to make objects, and we can relate objects to past events, to remember faces, for example. So we start to build on each simpler bit of information to create more complex information. And that's what's done in association cortices. Association cortices can be what we call unimodal, meaning that they deal with one sensory modality, or they can be heteromodal. They deal with multiple sensory modalities, such as vision and hearing. So we have primary and association cortices. You'll notice a big chunk of the frontal lobes is going to be heteromodal association cortices, because our conscious awareness involves putting together all sensory modalities, as well as our emotions and past events as well. Now, the location of these different primary and association cortices is really what gives all the different lobes their distinct functions. In the frontal lobes, we carry out what we call executive function, or kind of putting together our life. So here's an example of someone who has damage to his frontal lobes, and you can see how that affects him here.
Cancer and raising 7 children, Becky Harper thought that she was going to call it quits on her husband. He was apparently putting her down. He was emotionally attacking her, and she said, enough is enough.
Well, Becky and Jerry Harper told Fox 4's Neha Curtis they found out something else was Jerry act out something inside him they could not see. Neha's live in our Dallas newsroom. Tell us about it.
For years, a tumor was growing inside Jerry's brain, slowly pushing against his frontal lobe, the part of the brain that affects our personality and how we act. Becky says she didn't even recognize the man she married.
Do you want the Cavenders too?
No, thank you.
Okay.
Becky and Jerry Harper were on the verge of divorce after 18 years of marriage. Like walking on eggshells, because you never knew what was going to set him off. And it was like, no, I'm not living this way. No one had a clue what was behind Jerry's personality change, from someone who's kind and laid back to someone who snapped at anything.
I was getting more upset with guys at work. You know, I'm usually pretty easygoing at work, and I was, taking a lot of stuff out on them too.
Here, what you see is this area of whiteness.
Until one day when Jerry collapsed and doctors found out he had a non-cancerous brain tumor the size of a plum pressing against his frontal lobe.
The best analogy that you can draw to it is someone who's been at the bar a little bit too long and is a little bit free with their speech.
You just thought he was being a jerk. Just pretty much, yeah. Yeah, pretty much. One day, Jerry...
Moving behind the frontal lobes there, we have our parietal lobes. And what we need to think for our parietal lobes is somatosensory function, but also visual spatial organization. You know, when you think about how do we figure out where an object is, well, of course we look for it. So right now the camera is in front of me, it's to the right of me, now it's to the left. I'm not getting the sense that it's moving because I also know where my body is in space and I'm relating it to my body. So in order to actually localize the stuff that we see, we need to know where our body is. So it makes sense that you'd have somatosensory and visual spatial function in the same area because they're essentially the same task. So here we can see what happens with damage to the parietal lobes. We're going to see what we call hemi-neglect, where one half of the world is lost. The patient will just pretend like it doesn't exist. So this is a common task called the clock drawing task, where you ask a patient to just draw the numbers of a clock. We all know what a clock looks like, but it requires you to have a good sense of space.
Put the numbers on the...
Oh, how they go?
Yeah, where the numbers go.
1. Is that how it got chewed up? 2.
Yep.
3.
Now you'll notice here, she's gone ahead and skipped about half of the clock there. So 9, 10, 11 o'clock, those are all pretty close to the 6 o'clock position there. So she's put the numbers on, it's just the arrangement doesn't make sense. And this is because she has left neglect. So the left side of the clock doesn't really get filled in very well. So for parietal lobes, think position. Moving back, the back of the brain here, we have the occipital lobes. Damage to the occipital lobes is going to cause visual abnormalities. Now, where the damage occurs is going to determine what types of visual abnormalities. You might have cortical blindness where you see nothing. There might be a complete loss of color. And this is different from color blindness. This would be achromatopsia, where everything just looks black and white. This is different from color blindness, where we just fail to distinguish certain wavelengths from one another. So here we can see, this hypointensity back here, suggesting to us that there's damage in the back. Of course, the back would be the occipital lobe, so we should have some sort of visual impairments. Now, it's not in the primary visual cortex, so we wouldn't expect cortical blindness. We can still see all of the basic figures in the vision, but the association cortex is damaged, so that's going to impair our ability to create visual perceptions. And what our brain does whenever we have a lack of information, it just makes stuff up. It does this all the time. So we all have a blind spot that we fill in. Our peripheral vision is largely colorblind, but we don't perceive that. We fill in the details. When that happens, when we have a lot of details to fill in, well, it becomes a little wacky.
We're outside, and my friend Mary and
So
Jamie were
this is Charles Bonnet syndrome. You're going to find she's a little difficult to understand, but you'll get the gist.
Jamie going up in the air and back down again. And buses going up and down, and there was no buses. I know that there's nothing there. What I'm saying, there's nothing there. It's not real. Buses come along. If I'm standing waiting at a bus stop and I'm looking for a bus coming along the road, there's maybe no a bus coming, but I can see buses coming out of streets that don't come out of streets. Buses going up in the air. It's just because I'm maybe concentrating and looking for a bus.
So when we have damage to visual areas or auditory areas, and we have a lack of input in those sensory modalities, our brain just fills in the gaps, and so it can create visual or auditory hallucinations. The temporal lobes are going to carry out really three key functions: hearing, memory, and emotion. So language is of course going to be a big part of the temporal lobes, since language involves, of course, a lot of hearing. Language also involves production. So we're going to bring the frontal lobes in as well. So there's two areas: Broca's area in the frontal lobes, where we have motor function, and Wernicke's area in the temporal lobes, where we have auditory function and our memories. So it's where we make sense of the words. Here's an example of a gentleman with Broca's aphasia.
Now tell me what this thing was with your legs last week or week before. No good.
So you can see that he's struggling to put together more than one word at a time, or even coming up with one word. This is in contrast to what we see with Wernicke's aphasia.
What's this?
You know, the big, end of the football.
End of the football?
It influences the football right here. The cause that it comes in, see?
So this gentleman can produce language, it just doesn't make any sense. So in Broca's area, think broken Broca. You're unable to create language. Wernicke's, think wacky Wernicke's. You can make language, but it won't make sense to anyone. Hidden underneath our temporal lobes, we have two key structures, which through the magic of Panopto's errors are already on the drawing. We have the hippocampus, which is key for memory storage, particularly semantic memories, facts that we know, things that have happened in our lives. Just anterior to the hippocampus, we have the amygdala. The amygdala is often thought of as the site of negative emotion, of fear, of anxiety, but it does more than that. It's involved with any emotional computation. So here's the cerebrum, temporal lobes are highlighted here, and buried beneath that, so more medial to our cortex there, we have the amygdala and the hippocampus. We have an appreciation of how important the hippocampus is for memory function from patient HM. There are more modern examples, which we'll go through later, but patient HM was the first gentleman to lose his entire hippocampal complex on both sides of his brain. He had an intractable form of epilepsy that was unable to be controlled medically, so the only option was surgical intervention. And in the surgery, they removed his medial temporal lobes, so those parts that are hidden under the temporal lobes here, such as the hippocampus. The surgery was a success from the standpoint of controlling his epilepsy. The seizures were largely absent afterwards, but so was his memory. Patient HM from that point on was unable to form any new semantic memories. So he learned nothing from that point on. He could learn implicit memories, motor functions, for example, so how to move, how to navigate a maze by looking in a mirror, so carrying out mirror image movements. It's a little difficult to do, but with practice you can learn. Now, of course, he didn't remember having any practice with this. He just assumed he was a natural, I suppose. So when you remove the hippocampus, either surgically or in some cases through viral infection, you remove memory. We think of the hippocampus as being a site to hold memories while we determine, are they important? And if they are important, they'll gradually be moved to the cortex. And this is why patient HM still had his long-term memory intact. Everything that was already in the cortex, such as his name, events that occurred in his childhood or several years preceding the surgery, remained. The cortex was undamaged. The hippocampus was removed. So he no longer created any new memories. And he worked with the same set of researchers for decades and not once remembered them. Every day it was a new day. It was like they were being introduced for the first time. And in fact, when the researchers would go out into the hallway for a few minutes and come back in the room, they had to be reintroduced again. The importance of the amygdala is clear with animal studies, but we also have a patient, in this case SM, who was born with a genetic condition that caused degeneration of both amygdala. So here we can see the amygdala, here, there's the hippocampus very close to it. Down at the bottom we have patient SM. You'll notice the amygdala is largely absent here. Patient SM doesn't experience anxiety or fear because she lacks the amygdala. So temporal lobes are going to be important for auditory function, memory, and emotion. There's a lot going on in the temporal lobes. In the temporal lobes, we're going to receive some of that visual input. So from the occipital lobe, we can head on up to the parietal lobe to figure out where stuff is, or we can head down to the temporal lobes to figure out what stuff is. Different types of stuff will be remembered in different parts of the temporal lobes. For example, there is an area called the fusiform face area, which is where we figure out faces. When there's damage to the fusiform face area, patients suffer from what we call prosopagnosia, or face blindness. Here's an example here.
Meet Terry Sweeney, a professional photographer. She was a firefighter until an injury damaged part of her social brain, the circuitry she uses to recognize faces. The disorder is called prosopagnosia. For example, The person Terry has been photographing here is her mother. Now, because of her injury, Terry can't recognize any face, not even her own mother's. In this test, Terry takes a Polaroid picture of her mother.
Terry, you just took a picture of your mother. I want you to take a look at these and see if any of them are the picture you took or not.
Now, Massachusetts General Hospital researcher Dr. Nancy Edkoff shows Terry several Polaroids with only faces showing and asks Terry to pick out her mother.
Okay, if you had to guess, which looks to you looks like your mother? Just looking now, you can't tell which one of these. That is really look like your mother to you. Okay, if you just cut off the face, I just look at the clothes, I can tell you. Okay, do you want me to uncover them? Okay, why don't I do that? I'll uncover them so you can see what they're wearing and other parts of the picture. See, that's where it goes. The pink and purple shirt. The shirt.
So the fusiform face area is down here. So here we're looking at a reconstruction of the brain, right hemisphere, left hemisphere. So frontal lobes, occipital lobes, this is where we have our visual input, and then going down into the temporal lobes. Here if we look on the underside, we can see the bottom of the temporal lobes. We see this fusiform face area. It's in a slightly different spot on everybody, but it's always in the temporal lobes. When this is damaged, we become unable to see faces. The way that it's described by patients with this is essentially it's like looking at someone's elbow. It's very hard to tell one person's elbow from another. The thalamus, which is going to lie below our cerebrum, so all those different lobes we just went through. In thalamus, we should be thinking of grand central station. Everything goes through the thalamus to get to the cortex, and in fact, parts of the cortex talk to one another, in some cases by going through the thalamus. So the thalamus is in some cases a sensory relay. In other cases, it's almost like association cortex. Below the thalamus, we have the hypothalamus, which plays an important role in neuroendocrine function, or the release of hormones into our body. Below the thalamus, we get into our brain stem. So for midbrain, let's think of that as controlling the eyes, and we have a whole bunch of tracts that are very important to keep us conscious.
This patient has a right third cranial nerve palsy with complete right upper lid ptosis. His right eye abducts normally, but it does not adduct normally. It does not supraduct normally, and it does not infraduct normally.
So you'll notice here, great difficulties moving this eye. He also can't lift the eyelid at rest, which is why he's holding it up here. He requires a little help here, so the third cranial nerve is going to help keep our eyelid elevated, and it's going to perform essentially every movement of the eye except abduction. There's a little bit that the trochlear nerve does too, but most movements are going to involve the oculomotor nerve. It also has important parasympathetic function that may or may not be damaged. Here we can see what happens whenever we have damage to the cerebellum. So hanging off the back, we got our little brain. Here's a gentleman who has damage in the central portion of the cerebellum, the vermis. So here, he's suffering from balance issues. So when you damage the center of the cerebellum, you essentially cause a lack of coordination in the center of the body. When you damage more lateral regions of the cerebellum, you'll cause a lack of coordination in the limbs, or the more lateral portions of the body. Pons and medulla we're going to put together because they, for the most part, work together. The pons is a little above the medulla, both structurally and functionally. The pons is going to regulate medullary function. but they're going to work together to carry out vital tasks. Breathing, swallowing, cardiovascular function, for example. And when we have damaged these regions, it's more likely to result in death than damage in other regions. And if it doesn't, then you can be left with a severe impairment in function, because there's a lot of tracts that run through here. For example, the corticospinal tract. Our upper motor neurons, of course, have to pass through the midbrain, pons, and medulla in order to innervate the spinal cord. Now, the midbrain is where we create conscious arousal, though. This is what has that ascending arousal system to keep the cortex awake. So when we have damage here, we remain awake but unable to move. And this can create what we call locked-in syndrome.
Mike and Wendy Cubis, and memories of a more normal time. It was a couple of days after this holiday that Mike had a massive stroke. It left him paralysed, unable to talk.
He was 40.
He was very young.
Incredibly young. It was very bleak, really, apart from I had to carry on and the boys kept me going. They were only aged 12, 10 and 8, and I think the eldest one found it very difficult to cope with.
Mike has locked-in syndrome, but is able to communicate using his eyes.
Now you'll notice he can still move his eyes, because again we have damage below the midbrain. So he's able to be conscious and move his eyes. So he can use charts to try to convey his desires. And he still, of course, has desires. He still has his self. He still has his cortex intact, so he's completely aware of what's going on. He's just unable to move anything except for his eyes. For some folks, this leads to despair. For others, not so much. You can finish the video and find out. Mike seems okay with it. Not everyone is, though. So we got a few questions here that we're going to go over in class. Make sure you spend a little time and you can answer these. If you have any questions before then, always send me an email. Otherwise, I'll see you in class.